Hi students, welcome back to the Sound of Science. Today we're going to be talking about the genetics that's a little bit more complicated than the monohybrid cross. Just a quick reminder, we're going to be using the words genotype and phenotype. Remember, the genotype are the genes that are present. We usually represent those with letters. Phenotype is the physical characteristics, the physical traits that we can see. So we're going to talk about those, and we're going to talk about what we want to do when it is a little bit more complicated. For instance, we're going to start by talking about a dihybrid cross. Now, one of the things that Mendel came up with as part of his postulates, he is dealing with the law of independent assortment, right? That means that alleles sort themselves independently of each other. So just because I have blonde hair doesn't mean I will necessarily have blue eyes, right? And Mendel discovered that working with pea plants, that just because a pea plant was tall didn't mean that it was going to have green peas instead of yellow. Or just because a pea plant produced wrinkled peas didn't mean that it would necessarily be green peas. So once he figured that out, he needed a way to see probabilities with multiple alleles, because maybe he wanted to know, what is the probability of getting a green wrinkled pea? We're going to do a dihybrid cross. Your monohybrid crosses, mono meaning one, hybrid is just a type of cross. Your monohybrid crosses have 4 little possibilities that you can get between the parents. Dihybrid cross is going to give you 16 different possibilities to fill in. So it's going to be a little bit different. The ratios are going to come out a little bit differently, but it works almost the same way. We just have an extra step in the beginning. Don't let these intimidate you. They will look complicated and complex. Please don't become intimidated. You can do this. The first thing that we do, our first step, is actually to FOIL our parents' genotypes. So for an example, I am actually going to use these parents right here. They're heterozygous for both genes, and both parents match in that regard. So both are heterozygous for round peas, and both are heterozygous for yellow peas. So in this case, we're not going to have any green peas in the parents, and we're not going to have any wrinkled peas in the parents, okay? So it can be round or wrinkled, yellow or green. So because the parents are the same genetically, we actually only have to do one FOIL set. Normally you would FOIL both parents, but I did it this way just to kind of save time in the video, and because I really don't know that you need to see it 1,000 times. When we say FOIL, we mean FOIL exactly like you do in math. First, outer, inner, last. So you guys are going to be looking at your first alleles. So take a parent. I've moved it over here just so you can see it. First, big R, big Y, that's our first alleles. So we would actually write down big R, big Y. This is going to become important in a minute. Our outer is this big R and our little y. See, there it is. There's our outside. Our inside is little r, big Y. And of course, finally, our last are going to be this little r, little y. So there we go. So first, outer, inner, last, FOIL. Cool. So this is what's going to go on our Punnett square. So I'm going to go to the next slide. I'm going to show you what the dihybrid cross looks like. We are doing a cross of these 2 round yellow peas that are heterozygous. Hetero meaning one dominant, one recessive allele, right? So we're going to take these 4 that we made, and we're going to place them along the top and along the side. This is where the parents go. And remember, the FOIL was made of the parents. And then just like normal, you're going to drop and you're going to slide. And it might look a little different, but it's still the same process. So you're going to end up with 2 allele sets in each box instead of 1. That's also why we have 16 different probabilities or possibilities. So in this case, big R drops down, big R slides over. And so for this allele, this gene, the P is going to be round instead of wrinkled. It's dominant. Big Y drops down, big Y slides over. Again, this is a dominant trait for this gene, so the P will be yellow. And then we can do the same thing down here. So let's say that the big R is going to drop down here. And, little r drops down here. Big R is going to slide all the way over. That gives us a round P. Little y drops down. Little y slides all the way over. That gives us a recessive and a green P. So we talk about the ratio here. And sometimes when we talk about that ratio, it's this particular ratio of the two heterozygous alleles for each parent actually gives us kind of a special ratio. And that ratio is going to be 9 to 3 to 3 to 1. So what do I mean by that? Well, essentially what I mean is we're going to have 9 that are in our dominant category. So there's 4, 5, 6, 7, 8, 9. That means they're dominant for both. There's going to be 3 that are going to be dominant for one, but recessive for the other. So in this case, this one is going to be, and we'll just do a star, dominant for the first trait, but recessive for the second trait. And we've got 1, 2, 3 of those. And then this one is going to be recessive for the first trait, but dominant for the second trait. So I'm just going to do a circle. 1, 2, 3. And then our 1 over there, our last little 1, is going to be recessive for both. And so that one, I'm just going to put a little smiley face. There you go. So that's what we mean by 9 to 3 to 3 to 1. So now that we've done that dihybrid cross, let's talk about slightly more complicated genetics. We're going to begin by talking about incomplete dominance. So this is something that Mendel didn't really experience or didn't really address. And so I'm going to sort of explain it to you and explain how it's different. So in Mendel's world of peas, the pea plants were either tall or short. There was no in between. There was no medium plants. The flowers were either white or purple. There was nothing in between that. The peas themselves were either yellow or green. There was no yellow-green peas. And so for Mendel, that was straight dominance recessive. And it's really good for us that he was able to determine that. But unfortunately, that's not how all genetics work. Incomplete dominance just means that while we still have a dominant allele, in this case, a red flower, and a recessive allele, in this case, a white flower, the dominant allele is not completely dominant. It isn't able to completely hide the recessive allele. So when you have a heterozygote, which has both a dominant and a recessive, a little bit of the recessive is going to bleed through. This is a blending of the dominant and the recessive trait. And so incomplete dominance is a really special circumstance. And you are not going to be expected to identify which traits are incompletely dominant. But if you're given a problem with an incompletely dominant allele, you'll be told that, number one. And second, you'll need to understand that means they're going to blend together. And that's a really important distinction. Because in codominance, they don't blend. In codominance, this is an example where they literally just occur at the same time. So codominant alleles are the only alleles we're going to see that are going to be expressed with 2 different letters. Up until now, it's always been a dominant allele is given with a uppercase letter and a recessive allele is given with a lowercase letter. Codominance, it's going to be given 2 uppercase letters and they'll just be different letters. So in the case of these chickens, and this is a real scenario, I didn't make this up just because I like chickens. White chickens, white feathers on this particular breed of chicken are indicated with two dominant alleles that say have white feathers. Black feathered chickens have two dominant alleles that say have black feathers. If you have both a black and a white allele in a chicken, they're actually going to have speckled feathers. They're going to have some black feathers and some white feathers. And so you actually see this appearing in a couple of different circumstances. The most often that come to mind are going to be in these chickens. So you get speckled black and white chickens. You also can see it in cows and horses. And it's not black and white, but it's actually red and white. And so the cows and the horses appear to be roan is the name of the color. And they're just sort of dappled red and white. And sometimes their fur sort of blends so well together, they almost look pink. But again, this is not a blended situation. This is where individual cells have different hair colors or different feather colors, and you see both. Your indicator for codominance is going to be the presence of two different letters. Anytime you're given two different letters, that is codominance. To complicate things a little bit more, you also can have multiple alleles controlling a trait. So instead of having just black feathers or white feathers, instead of having just red flowers or white flowers, instead of just being tall or short, in this case, you actually have more than two options. So for bunnies, your wild type, that's your brown bunnies. You also can have the chinchilla type of fur pattern. You can have a Himalayan type fur pattern and an albino type fur pattern. And these are all, there's not a codominance here. There is a recessiveness here, and they blend in different ways, or they show up in different ways. But essentially, wild type is dominant, and then the recessives can all be found with these three different allele types. So it just depends on whether you get a recessive chinchilla type, a recessive Himalayan type, a recessive albino type, or some combination thereof. So this is a case where we see multiple alleles. Humans have this as well. We actually have multiple alleles for some of our characteristics, just like rabbits do. And to get even more complicated, let's talk about polygenic. Polygenic just means multiple genes. These are traits that are controlled by more than one gene. Most of our traits actually fall into this. So height for humans, it's actually like 9 different genes that controls how tall we are. Parakeets, they have multiple genes that determine parakeet feather types. And so that's why parakeets are given as this example. But regardless, these are all polygenic traits. These are traits that are controlled by multiple genes. So finally, let's put all this together. Human blood types. Human blood types have a little bit of almost everything, actually. They have recessive alleles. That would be the type O. They have multiple alleles, A and B, as well as the recessive. And they have multiple genes. They're polygenic. Letter antigens versus Rh factors. So blood types really combine all of this, which is why we like to talk about them. Not to mention the fact that you're the owner-operator of a human body. You should really understand how blood types work. So blood types appear in 8 different varieties. You can have O negative, A negative, B negative, AB negative, or O positive, A positive, B positive, or AB positive. Yikes, that's a lot of blood types. But it's really kind of simple when you break it down. The first thing I want you to understand is that A and B are codominant. Just like co-parenting means they both show up. So this is not a blended case. This is a both will be there case. Okay? So A and B are codominant. O is recessive. So if you have a recessive allele here, you're not going to have any antigens. Well, if you get 2 recessive alleles, you're not going to have any antigens. If you get an A allele, you'll have A antigens. If you get a B allele, you'll get B antigens. And if you get an AB allele, you'll get both. You can have A and B. The other gene is that Rh factor. It sorts completely independently of the A, B, and O gene. So in this case, if you're positive, you're going to have Rh factors present. So for instance, A positive has both A antigen and Rh factor. So on one allele, it has A, and on the other allele, it's going to be the dominant Rh. Okay, Rh factors are straight up Mendelian, dominant, or recessive. So, what we see here, because it's looking at two different alleles, and because some are codominant and some are not, that's what gives us our 8 possible blood types that we can experience. The really cool thing is that your body is designed so that it can receive blood types or blood from somebody who has the same antigens as you, or at least doesn't have any antigens that are different. So, for instance, if somebody is A positive, they can actually get blood donated to them from somebody who's A negative because they have the A antigen, somebody who is O positive because they have the Rh factor, and somebody who's O negative because they don't have any antigens that we don't see here. And of course, somebody who is A positive. So you can see that. Take a minute and try to figure out who can donate to who and who can't. I'll give you a hint. O negative is the universal donor. That means they can donate to everybody. AB positive is going to be our universal recipient. They can receive blood from everybody. So we're going to do some more work with blood types. I'm going to actually show you a blood type lab, assuming that we can stay in school long enough. And hopefully it'll make a little bit more sense after practice. So hope this is all kind of congealed a little bit for you. It hasn't been too complicated or too confusing. We're going to practice with this once you guys get a chance to view this video. And as always, if you didn't take your notes, go back and take your notes. Make sure you've got everything written down that you deem is important. Definitely focus on the different type of genetic inheritances that we can have, and just be prepared to be able to do a dihybrid cross. So good luck to you, and until next time, see you then.